Choosing the right protein expression system can make or break a project. Today we'll walk through the main platforms and when to use each one. Target, timeline, and budget. Those three parameters guide the choice. First up, bacterial expression. Meet E. coli and Bacillus, the workhorses of the protein world. They grow fast, reach cell densities that push yields up to 10 grams per liter, and run on the cheapest media you can buy. If your protein doesn't need fancy post-translational modifications, or if you can refold from inclusion bodies, bacteria are usually the fastest and most cost-effective route. They're ideal for vaccines, industrial enzymes, and structural biology targets under about 150 kilodaltons. Next up, yeast, both Pichia pastoris and Saccharomyces cerevisiae. These microbes give you eukaryotic perks without breaking the bank. They secrete proteins straight into the broth, making downstream purification easy, and they add high mannose N-glycans plus reliable disulfide bonds. Yields rival those of bacteria. Timelines shrink to 4 to 8 weeks, and scale up to 5,000-liter fermenters is routine. Choose yeast when you need secreted enzymes or antigenic proteins up to about 200 kilodaltons. Now enter the insect world. Spodoptera frugiperta SF9 or SF21 cells powered by baculovirus. This system excels at folding large, complex proteins and inserting authentic phosphorylation sites or complex N-glycans. It's the go-to for kinases, virus-like particles, and tough membrane proteins that would choke other hosts. You'll see lower titers, 0.1 to 0.5 grams per liter, but the fidelity you gain is often worth it. 5 to 7 weeks from gene to purified protein is typical, and bioreactors up to 2,000 liters are readily available. For true human-like post-translational modifications, turn to mammalian cells, CHO or HEC 293. These cells add proper sialylation, galactosylation, and every other PTM your therapeutic antibody demands. Transient transfection gets you research-grade material in 3 to 5 weeks, while stable pools or clones deliver multi-gram cGMP batches within 3 months. Costs are the highest, but regulatory agencies know and trust these cells. If you're developing a biosimilar or any biologic destined for the clinic, this is where you land. Finally, cell-free systems, E. coli, wheat germ, insect, or human extracts, tear down the walls of living cells. Reactions run in hours, scale from 96 well plates to liter volumes, and never worry about host viability. That means toxic or membrane proteins can be produced at microgram to gram scale without cellular stress. Human and insect lysades even provide native glycosylation and disulfide formation. Use cell-free when speed is critical, the protein is toxic, or you need rapid antigen supply for pandemic response. It's the fastest route from gene to detectable protein when timelines matter. So how do you pick? Start with your protein's needs. Does it require human-type glycosylation? Go mammalian. Is it a secreted enzyme for industrial use? Yeast will do. A kinase or VLP. Insect cells shine. Toxic or very large? Cell-free is your friend. Then weigh your timeline and budget. Bacteria are cheapest and fastest for simple targets, but the cost climbs as you add PTMs and regulatory compliance. Expect costs and timelines to scale with fidelity requirements. Tell us these 3 parameters: target, timeline, and budget, and our scientists will map them to the optimal platform at no charge. We pair platform expertise with supply chain reliability and QC oversight. Tip: try bacterial expression first for simple constructs, then move up to eukaryotic systems if you hit folding or PTM roadblocks. Optimize construct design, signal peptides, codon usage, and tags can change outcomes. Plan for downstream purification and analytical assays before scaling. Structural biology targets under approximately 150 kDa often live happily in bacteria. Industrial enzymes and large-scale antigen production commonly favor yeast or bacteria. Therapeutics and clinical candidates usually require mammalian hosts and regulated processes. Need help choosing the right system? Submit your target, timeline, and budget and get a tailored recommendation. Our scientists and 24/7 technical support will guide the plan. In short, pick bacteria for simple, cheap, fast production, yeast for secretion and scale, insect for complex folding, mammalian for true human PTMs, and cell-free when speed or toxicity is the limiting factor. Remember, expression is iterative. Screen multiple systems early to save time later. Work with partners who provide batch-consistent reagents and QC oversight. Good planning turns risky projects into reproducible results. Thanks for watching. Protein expression choice is a strategic decision, not just a technical one. Creative Biomart offers technical support to help your project succeed. Tell us your target, and we will advise the optimal platform.